Hi, everyone. Thanks for joining. I'm very excited to introduce Stuart Hameroff, Emeritus Professor at the University of Arizona, Director of the Center for Consciousness Studies, and attending anesthesiologist at Banner University Medical Centers. Dr. Hameroff, in collaboration with Nobel laureate Sir Roger Penrose, is the developer of the controversial orchestrated objective reduction quantum theory of consciousness in microtubules, which he formed with Penrose in the early 90s. Since then, he has worked to test and validate Orch OR, as well as help pioneer transcranial ultrasound to treat mental and cognitive disorders. Dr. Hameroff also co-founded the University of Arizona Center for Consciousness Studies and directs its annual conference, The Science of Consciousness. So Stuart, welcome, and thank you so much for joining us today. Please take it away.
Thank you, Paige. Thank you for inviting me, and thank you all for being here, wherever you are. And my topic is the quantum underground, where life defeats decoherence. Anybody in this field has heard a million times, the brain or biology is too warm, wet, and noisy for delicate quantum effects. You need absolute zero temperature. I don't think that's true. And the beaker you see with water with some oil poured into it illustrates a point I want to make. The oil and water are separate. Oil and water don't mix. And I think that's kind of the key of quantum biology. Uh-oh. My slide's not advancing.
Try the...
Oh, wait. Okay. There's a way to do it. I forget. Okay. Okay. So the real title of the quantum underground should be the Meyer-Overton quantum underground. And you'll see why. Meyer-Overton, it comes from the anesthesia research world. So let's start with asking, what is life? I'm saying that life may be related to quantum biology and vice versa. And it's hard to even define life. And there are functional approaches. For example, the famous biologist Lynn Margulis-Sagan described life as its function: self-organization, metabolism, growth, adaptation, response to stimuli, replication, reproduction, and evolution. But these occur in computer programs, video games, and weather patterns, such as the great red spot on Jupiter that you see behind me. And so that is not really an explanation. It's more of a functional description. And then there was vitalism that came along in the 19th century, in which there was proposed to be a unifying energy field, a life force, or elan vital pervading living systems. And this was based either on electromagnetism or on mysterious forces outside science. And when molecular biology came along, vitalism became taboo. It didn't seem to be necessary. I'm not so sure about that. So there's also complexity, various forms of nonlinear dynamics, chaos theory, scale-invariant logic, information flow, emergence, self-organized criticality, et cetera, by a number of authors, including some good friends of mine, in various ways. But again, these can all happen in non-living systems, so they can't truly be the essence of life, I don't believe. And finally, Erwin Schrödinger, in his famous book, What is Life, in 1935, suggested that life's unitary oneness derived from quantum coherence, and that memory was stored in aperiodic crystal lattices. And I think this was right, actually. But everybody said and continues to say, biology is too warm, wet, and noisy for functional effects in quantum systems easily perturbed by thermal decoherence. But is it? We got to remember that biology is highly heterogeneous. It's not a monolithic slab of one type of material. So, for example, if you look at a cell, and this looks like a bacterial cell, it's 70% water, 30% chemicals, and those are divided into proteins, small molecules, RNA, phospholipids, DNA, polysaccharides. So highly heterogeneous in that sense. And if we actually look at it, in this micrograph showing a cell, an immune cell on the left and in the blue, and then a blood vessel, a capillary or extracellular fluid on the right in the green, bounded by separated by a membrane, and the cell on the left is squirting immunoglobulins or something into the blood, and which is basically liquid. And in the cell itself, you see cytoskeletal structures going from kind of diagonal up towards 10 o'clock. You see one microtubule, the large object, and then crisscrossing, you see actin filaments. So these are part of the cytoskeleton that I'm very interested in. Here's another picture of that, just looking inside, might be a neuron. You see two parallel microtubules crisscrossed by actin and neurofilaments and some other things floating around. And if we look just at the cytoskeleton in a cell between two membranes, we can see the microtubules in the green, the intermediate filaments in the blue, and the microfilaments or actin in the purple. And so the question, one question is, and I'm an anesthesiologist, as Paige mentioned, and I've been doing it for a long time, and I take care of patients for surgery. I put them to sleep. I take away their consciousness, and then wake them up at the end by removing the anesthetic. And so the question that anesthesiologists and pharmacologists are concerned with is, if we give a drug, whether we give it by somebody takes a pill and it gets absorbed in the stomach, or intravenous, or injection by intramuscular or sub-Q, or inhalation, breathing it and then crossing into the blood in the lungs, how do we know where it goes? How does it know where it goes? How's that controlled? And the answer is by its solubility in a particular phase or compartment. So here's a different way of looking at a cell or the body or the brain or any biological tissue in terms of its solubility phases. And if you look at this triangle, the main axis is polarity, going from lower right to upper left. So the more to the upper left, the more polar. And you can see water, number 26, is just below the arrow off to the left. And that's more and more polar as you go in that direction. The other direction is nonpolar and oil-like. And it turns out that those are very, very important. Now, most drugs are polar. That is, they have charges. And they're electrically charged. They're soluble. Because of that, they're soluble in blood and water because they can form hydrogen bonds with water. And they seek oppositely charged receptors on membranes with matching lock and key conformations. So imagine the key and the lock have, one has positive charges and the other has negative charges. This molecule has a bunch of charges that are opposite to where they are on their binding site. So it's going to be attracted and it's going to lodge there and kind of stick, both by charge interactions and also by conformational, this matching lock and key. And this is where all the specificity in drug design comes from. And there's probably an infinite number of structure activity relationships. But what about uncharged nonpolar molecules with complete outer electron shells, like the inert gas xenon? So xenon is an atom. It's an element, and it's got a completed 8 electrons in its outer shell. I think it's got 54. I forget. Its atomic weight is 54, or something like that. But the outer shell is inert, or it's inert because the outer shell doesn't have any spare electrons or any holes for charges. So it's inert, right? Though it's chemically inert, xenon has a particular effect when inhaled into the lungs and through the blood, reaches the brain and its nonpolar binding regions. So that's the key, that these nonpolar drugs go to nonpolar binding regions. And like other nonpolar inert gases shown here, a few others, xenon is an anesthetic, selectively blocking consciousness while sparing non-conscious brain functions, which continue under anesthesia. The brain's quite active under anesthesia. We use evoke potentials, evoke responses from the back of the brain to the front of the brain. Excuse me. In anesthetized patients who are unconscious, for spinal cord surgery to make sure that the signals are getting through. We have somebody stimulating the feet or the hands and make sure they get through. If the surgeon compromises the spinal cord, they go down and he does something different. So the brain is still active, and all these drugs affect, strangely enough, is consciousness, despite the fact that they're very non-specific in their binding, which I'll get to in a second. So let me talk about anesthesia, which has been my day job for, I hate to even tell you how long, a long time, over 40 years. And in the year 2000, this book came out about the greatest inventions of the past 2 millennia. And I wrote the chapter on anesthesia, which, as you can see, is at the top of the list, alphabetically at least. And what I said basically was, imagine if anesthesia didn't exist, we would go around in risk or worry of our appendix bursting or breaking an arm or whatever. And then what? Imagine a world without anesthesia. It's actually kind of hard to imagine. But it would be it'd be full of trepidations and pain, I would suspect, unless something else came along. So in the 19th century, a group of gases were found which, when inhaled at low concentrations, caused giddiness and euphoria. And here are a few of them. So ether frolics. People figured out if they breathed a small amount of diethyl ether, they became very happy, high, and had a lot of fun dancing around and so forth. And similarly, laughing gas, nitrous oxide, was used for partying. And you can see at the top, living made easy. And I think at the bottom it says a treatment for scolding wives. I'm not going to comment on that. I don't know if you can see that caption or not. But nitrous oxide and ether at low doses both caused euphoria. And typically, a lot of drugs actually are biphasic. At low doses, they cause one effect, and you go into a little bit higher and you have the complete opposite effect. It may be, in some cases, agonizing the receptor and then blocking it, but there's no known receptor yet for anesthesia. At higher concentrations, anesthesia rendered patients unconscious and unresponsive, enabling modern surgery. And this is a famous depiction of the first public demonstration of ether as an anesthetic at the Mass General Hospital in Boston in 1846. And I've been doing anesthesia a long time, but not this long. I wasn't there. I came along quite a bit later. But the anesthetic gases have varying chemical structure. There are ethers, there are halogenated hydrocarbons, there's the inert gas xenon, there's nitrous oxide. They have different structures, but the same effect. And that is because apparently that they each have a nonpolar surface, shown here in gray, of complete electron outer shells, which bind by very weak van der Waals quantum dipole coupling forces. So as I told you before, xenon is inert, and all of these gases are basically inert, but have a profound effect of selectively blocking consciousness. Oh, this isn't going to work. Okay, this is supposed to be an animation. This is neon, another inert gas, which the electrons in one repel the electrons in the other. And so each forms a dipole. It's called an induced dipole. So you can imagine all the electrons in one neon going to the lower left and the other one to the lower left. And then they attract because they're like two little bar magnets. And then they oscillate back and forth. Sorry, I can't get this to run in this setup. But anyway, an amazing fact, still amazing to me after all these years, is that anesthetic gases immobilize all animals studied. Mammals, amphibians, insects, fish, worms, reptiles, and even plants, like phototropism following the sun. That's blocked by anesthesia. Venus flytrap, that's blocked by anesthesia. And for each gas, each gas has the same average dose called the minimal alveolar concentration, or MAC, found to immobilize all animals. And what they did was they would take a mouse and clamp their tail, and they're going to move. Then they started giving them anesthesia gas, and they found the concentration of gas where half of the mice would move and half of them wouldn't, or the writing reflex, or various surgical stimuli. So MAC is the ED50, the 50th percentile concentration, where half the animals will respond or half humans will respond to a surgical stimulus. And so, by the way, anesthesiologists always err on the side of giving a little bit extra, because we don't want half of our patients moving or being awake, certainly. So if you go to like a standard deviation above, you're pretty good. Plus, we have other better good signs for early warning. But again, each anesthetic has the same MAC for all animals studying, suggesting that there's some common mechanism in all animals and some common mechanism for anesthesia, erasing consciousness, and therefore, I would say some common mechanism for consciousness. That may be obvious, I'm not sure. Anyway, this was an enigma, and scientists around the turn of the 20th century sought a common property among these disparate chemical structures. And they first determined potency for each gas in immobilizing animals and testing solubility of each anesthetic in various solvents. So they were looking for the solubility of the anesthesia to find out where they're acting in the brain, and therefore maybe where consciousness is coming from. And after a long time of work, these two guys, Hans Meyer and Charles Overton, Meyer in Germany and Overton in the UK, found the same thing, that the potency for every anesthetic correlated with its solubility in olive oil, of all things, which is a highly nonpolar, oily material. It's delicious on all kinds of things, and you can cook in it. But you can see it doesn't really have any charges on it except that one hydroxyl, and it's got aromatic rings and on both ends, or at least one end. So this was the solvent that best matches the region in the brain where anesthetics act, and therefore consciousness might arise. So they then, combining the two of their work, they then created this amazingly straight line over many orders of magnitude of the correlation between the potency of the anesthetic on the y-axis, which is in MAC. So the lower on the y-axis, the more potent the anesthetic. And on the x-axis, I don't know if you can see that, it's cut off on my screen, but it's the oil gas, the olive oil gas partition coefficient. So the more to the left by methoxyfluorine, the more potent, the more soluble the anesthetic is in olive oil. And as you go upward to the right, it becomes less and less soluble. So nitrogen is an anesthetic, but at like 15 atmospheres. You'd have to pressurize 15 atmospheres, and that would have a slight effect. And xenon, which I mentioned earlier, is just under 1 atmosphere. So to use xenon, you can only use it as a partial anesthetic, or you have to use it at high pressure, so you can give more than 1%. Whereas methoxyfluorine, you only need 0.25%, 1/4 of 1%. So this tremendous range in potency correlated precisely with solubility in olive oil. The more soluble in olive oil, the more potent the anesthetic, and the less you need to anesthetize a human, a horse, or anything. So that is the Meyer-Overton correlation, it's called, between potency and anesthetic potency and solubility in olive oil. And this is the basis for what I call the quantum underground. Actually, Travis Craddock and I and our colleagues wrote a paper about this a few years ago, the quantum underground. So if we look back at the solubility phases of the human body, we saw that the polar molecules are going to go upward to the left, but the anesthetics are going to go down here. And I'm using Bing to imply consciousness, which I use in my talks often. I didn't use the setup slide for that, but that just means that's where consciousness is. It doesn't explain what it is. And it correlates with dimethylbenzene, methylbenzene, and benzene. So basically benzene, and which is aromatic rings, and the basis of what goes in our gas tanks, and basically all of life, actually. It's the basis for organic chemistry. So when I was in undergrad, I was told, if you want to go to medical school, you better ace organic chemistry. And I actually enjoyed it quite a bit. And you learn about the orbitals of the electron orbitals around atoms and molecules. And in particular, hydrocarbons and aromatic hydrocarbons. So in the, I think it was the 1700s, they knew about, chemists knew about hydrocarbons as chains of carbon and hydrogen, each carbon having 4 bonds. And they knew about alkanes, which had the formula CnH2n plus 2, because they were a linear chain of carbons with hydrogens, and alkenes, which were CnH2n, because they had 1 double bond. So they had 2 fewer hydrogens because of the electron bond. But they also knew about an oily flammable molecule or compound with the formula of C6H6, which didn't match either of these formulas. So one night, German chemist August Kekulé dreamt about the hydrocarbons, the linear alkanes and alkenes, as snakes. And in his dream, one snake swallowed its tail, which is mythically called the Ouroboros. And he woke up and said, aha, benzene, C6H6, is a ring. And the snake had swallowed its tail. And he turned out to be exactly right. Benzene has 6 carbons and 3 extra electrons that are shown on the right as these extra lines. And these 3 delocalized or pi resonance orbital electrons form pi resonance clouds, which are conducive to quantum optical effects. So here you see the pi orbitals for the individual carbons, then they coalesce into 2 rings and then into 1 electron cloud, enveloping the whole ring. And now if you had bulk benzene, it gets oily, and I don't know what happens then. But if you have these in arrays so that they're separated, like in graphene or fullerenes, then they have particularly interesting quantum properties. They have quantum dipole oscillations, spin transfers, excitons, photons, fluorescence, phosphorescence, and superradiance. These are all quantum optical effects because you can stimulate the electrons to excited states within these electron clouds. So for example, in tryptophan fluorescence, you take a protein that has tryptophan, the aromatic amino acid with the indole ring, the 5 and the 6, and you hit it with, say, 280 to 300 nanometer light, and you get an excited electron state. And after a short period of time, it will fluoresce and emit at about 340, a lower energy photon. And the top 3 step downs, that time can vary depending on how long it stays in these are quantum states, it looks like. And so the longer it stays up there, the longer it's in a quantum state. If it lasts longer, it's phosphorescence. It can also have what's called superradiance. So this is basic photo excitation, fluorescence of aromatic rings. Now, it's also known that if you take this was done in 2003, 20 years ago now, at UC Santa Barbara, where they took quantum dots, the green dot and the green dots and the red dots are quantum dots, and they're connected by benzene rings. You can see the benzene rings connecting the green and the blue, the green and the red. And if you put quantum spin into the system, the quantum spin is transferred from dot to dot through the benzene ring. And it turns out that this process is actually increased when you increase the temperature. So the idea that that temperature is the heat and thermal vibration is the enemy of quantum is generally true, but not necessarily. And in this case, at least for spin, it actually increases the quantum transfer. So that's promising. Now, if you just take the 2 benzene rings by themselves, and they will attract each other, as I said before, the electrons in one will induce a dipole in the other. So you have 2 dipoles which attract, form like little bar magnets, and then they oscillate back and forth in terahertz. So if you have these arrayed properly, you'll get terahertz oscillations out of these aromatic rings. Now, again, if you have it in bulk in your gas tank, then this is not going to happen. But if it's in a proper array, like in a periodic lattice, like Schrodinger said, then you can get oscillations and even quantum effects. And to make it even more interesting, I think, it turns out that anesthesia acts by blocking these dipoles by the same type of van der Waals oscillations, except they're called dipole dispersion forces, and block the oscillations. So you might wonder, and I did for many years actually, why the anesthetic doesn't just join in the oscillation, why its electrons don't just go along for the ride and oscillate back and forth. It turns out they don't, because there's something different about anesthetics, and I'll come to that a little bit later, because we actually figured that out in a study published in 2017. But the point I want to make here is that the anesthetic forms a similar van der Waals dipole coupling and blocks the oscillation, the terahertz oscillation. Now, this is similar to what's called Frohlich coherence. And Herbert Frohlich was a biophysicist who in the 60s and 70s proposed a feasible quantum state in living systems, kind of like a Bose-Einstein condensate. He said that coupled dipoles in nonpolar regions, for example, aromatic rings, that were in geometric arrays, so they had to be periodic, just like Schrodinger said, and in a common voltage, so they're all in the same voltage field, would oscillate coherently in terahertz, gigahertz, or megahertz in geometric lattices, and the frequency depending on the amount of mass that was coupled and moving. And I actually met Frohlich. He was quite a character and a brilliant guy. And I think this was an amazing suggestion that is still being talked about in various ways. Should also mention that psychoactive molecules, the neurotransmitters dopamine and serotonin, the pleasure molecule and the mood molecule, and psychedelics all have aromatic organic rings. And in the case of serotonin and DMT and psilocybin, they have not just the benzene ring, but the indole ring of the 5 and the 6. And this is also in the aromatic amino acid tryptophan, which I think plays a very important role. So this suggests that maybe this has something to do with consciousness, since these drugs tend to enhance consciousness. I should also mention that the psychedelics, and there's been a huge amount of work on psychedelics in the last number of years, have always been said to act on 5-HT2A receptors on the membrane surface, particularly in pyramidal cells near the soma. And it turns out just in this past year, it was discovered that, well, maybe it was known before, I didn't know it, that there are 5-HT2A receptors inside the neurons, inside the pyramidal cell, associated with microtubule associated protein. So as part of the cytoskeleton. And that psychedelics apparently promote neuroplasticity and are what Olson called psychoplastogens. They actually promote growth of neurites and sprouting and creating new synapses. So and it turns out they probably do that directly in the cytoskeleton. At least they are able to bind there. So and then we have amphipathic biomolecules like dopamine, like serotonin, like tryptophan and so forth, which have an aromatic ring on one end, which is nonpolar, and a polar tail on the other. And what happens is that the nonpolar rings attract, and if these are amino acids, for example, they attract and coalesce together, and the polar ends stick out to deal with the aqueous environment. And these are called micelles, which are kind of a progenitor of actual biological cells, described by Haldane and Operin back early part of the 20th century. So the aromatic amino acids coalesce, couple, and oscillate collectively in what are called hydrophobic pockets inside proteins. And when I was first learning about how anesthesia worked, the protein theory came along, and people called these regions of lipid-like regions inside of proteins hydrophobic pockets, meaning water wasn't allowed. Oil and water don't mix, so the oil-like benzene got there first and the water was kicked out. So this is a hydrophobic effect. So there's no water in there, so it's not wet, obviously, in case you're worried about too warm, wet, and noisy, the interiors of proteins. Also, lipid bilayers form in this way. The aromatic rings, the nonpolar aromatic rings coalesce in the middle which is hydrophobic, and the polar tails stick out outside the cell and inside the cell. And also in the pi stacks in nucleic acids in RNA and DNA stack up and form a continuous pi electron resonance column, you might say. And people have postulated quantum effects in DNA and RNA through this pi stack. So there's this these aromatic rings kind of isolated from the water in proteins, lipids, lipid membranes, and nucleic acids. And when Travis Craddock and I were thinking about this, we said, well, gee, it's in proteins, lipids, nucleic. That's pretty much all we got. And therefore, we decided, well, there are these quantum regions everywhere in a cell, not everywhere, but interspersed here and there inside these molecules. So that's where we came up with the quantum underground name. Now, where do anesthetics work? So I started studying how anesthesia works when I got into anesthesia because I was interested in consciousness. And because the Meyer-Overton olive oil solubility was lipid-like or oil-like, not charged, people thought, well, it must act in the membrane. And at that time, they didn't know about the receptors in the membrane. It was just lipids. And so they thought, well, the anesthetics get in there and expand the membrane, and that gums it up somehow, so it can't be excitable, not realizing yet that it was actually the proteins and ion channels. So that was the first theory by Seaman and Roth, I think, from University of Calgary. And then in the 60s and 70s, proteins, it was discovered that proteins were in membranes, receptors, ion channels, found to perform functions essential for neural activity. So it was the proteins that were accounting for the excitability. So then what? How did if the anesthetics were lipophilic and like to be in the lipid part of the membrane, how did they affect the proteins? Well, this fellow Jim Trudell at Stanford, who I knew very well, very good guy. He came up with this theory called Trudell's lipid phase transition, where the anesthetic. So at the top, you see a sodium channel, which upper left is closed, and upper right is open, and sodium goes through and the membrane depolarizes. Well, lower left, he's got anesthetics in the lipid phase
extrinsically compressing the ion channel, not allowing it to open. So this was his theory for how anesthesia acts. But it turns out that a slight temperature, he was saying the anesthetics disordered the lipids, and that didn't allow the protein to expand. But it turns out that a slight temperature change of 1 to 2 degrees would have the same disordering, and we didn't lose consciousness with a slight fever or getting slightly cooler. So this kind of fell by the wayside, especially when it was discovered in the 80s by Nick Franks and Bill Lieb in the UK. The anesthetics act directly on proteins in the hydrophobic pockets where these aromatic amino acids are. And they did it with a membrane-free system, luciferase, which gives off light with ATP. And they found that if with enough ATP, the light emission was inhibited by anesthesia, again, according to potency, kind of like a Meyer-Overton correlation. So they basically found a Meyer-Overton correlation on one protein. And that was really illuminating, because it meant that anesthesia was probably working on a protein, maybe one protein, maybe several proteins. So, but not luciferase, because we don't glow in the dark, at least most of us don't. And so people started to look for membrane proteins, ligand-gated ion channels, for example, became the presumed sites of anesthetic action. And anesthetic gases do indeed bind to membrane receptors for serotonin, glycine, acetylcholine, and GABA. And these became the favorite candidates. And the anesthetics do bind there. But experiments produce conflicting results. Some anesthetics open a channel, others close it. Not all anesthetics bind to any one receptor. And if you believe in a unitary mechanism of anesthesia and a unitary mechanism of consciousness, this just didn't make any sense. And the first rebellion against the membrane protein probably came from Alex Evers and John Steinbach, who wrote this editorial in the journal Anesthesiology, commenting on two papers that had completely opposite results. And their title was Double-Edged Swords: Volatile Anesthetics Both Enhance and Inhibit Ligand-Gated Ion Channels. The papers show that volatile anesthetics enhance and inhibit acetylcholine receptors, and they enhance inhibitory, but inhibitory at high doses. At low doses, they enhance; at high doses, they inhibit. So this didn't make any sense. And there followed a series of papers, all of which were very negative against anesthesia acting on acetylcholine receptors, 5-HT, opioid, adrenergic, potassium channels. They did not mediate the immobility effect. They didn't know that the animals were consciousness. So immobility means apparent unconsciousness or unresponsiveness. And GABA receptors didn't do it. So everybody was kind of at a loss. And then finally, the leader of the field of anesthesia, Ted Eager, who had led the charge, the investigation of membrane proteins for 15, 20 years, and along with a bunch of luminaries, people, big time names in the field of anesthesia mechanism research, came up. Pardon me?
Oh, okay.
Sorry, Stuart. I'm sorry, I muted you accidentally. I was trying to mute the voice that we heard.
Okay.
Sorry about that.
Okay, thanks. So anyway, Eager and these luminaries in anesthesia research came up, finally said, is a new paradigm needed to explain how inhaled anesthetics produce immobility. 2 decades of focused investigation have not identified a channel or receptor able to mediate immobility. All these receptors appear to be irrelevant. Furthermore, no combination of these channels seems sufficient. So this was quite a blow to the people who believed in membrane in receptor mediated anesthesia and also consciousness. So what was the alternatives? Well, actually, there were alternatives all along that they hadn't considered. For example, in 1846, Claude Bernard, a famous biologist, showed that cytoplasmic streaming in amoeboid slime mold cells, that they flow along by like amoeba, was reversibly inhibited by the anesthetic chloroform. If you just put the amoeba in a chamber with chloroform, it stopped moving, and if you blew off the chloroform, it resumed. And later, Seifert and Christensen showed the effect was directly mediated on cytoplasm, not via membrane proteins. So this was a direct effect on cytoplasm, and particularly cytoskeleton, which is responsible for movement. Actin does the flowing, but microtubules pick the direction. And then in 1968, and this was the paper that helped me decide to go in anesthesia, because my future chair knew this guy, John Nunn, knew I was interested in microtubules and consciousness and anesthesia. And he handed me this paper, Effects of General Anesthetics on Microtubules in the Lancet, a possible mechanism of anesthesia. And Allison and Nunn, and I met John Nunn a couple of times, very interesting character, studied these little organism called actinosphaerium, which are had these spikes coming out of them called axonemes. And if you looked at the cross section of an axoneme shown below, you saw this double spiral of individual microtubules. Every one of those little circles is a microtubule. And when they exposed it to halothane, the axonemes just disappeared. They just depolymerized and retracted. And so the halothane caused the microtubules to depolymerize. And so they came out in Lancet and said, well, anesthesia happens when you depolymerize your microtubules. But this required 5 Mach to depolymerize them. So 5 times the amount needed to put you or I or any animal to sleep was required to depolymerize your microtubules. Now that's good news, because we don't want to depolymerize our microtubules. And in fact, too much anesthesia can cause problems along these lines by probably causing depolymerization. Nonetheless, it did show that anesthesia acts on microtubules, binds to them, which was verified later. So at that point in time, in 2008 or so, anesthesia research faced a fork in the road, quite literally. Continue with membrane proteins, cytoskeletal proteins, or something else. And they kind of went something else and went back and looked at lipid theories, or just kind of ignored the whole thing, or went back to membrane proteins pretending there wasn't a problem. Nonetheless, evidence continued to accrue for effects on microtubules.
the Hameroff lab at Penn, and I think Paige said you went to Penn, right? Anyway, they did the best work beginning in 2008, right after the Eager paper came out. And they did it systematically. They said, okay, we're going to find out where anesthetics bind. And they used immunolabeled halothane and found 70 different proteins in brain neurons bind anesthesia, half in membrane and half in cytoplasm, including tubulin, the component protein of tubulin. And then they did genomics and proteomics, and they looked at the gene expression of these different proteins. And only 3 of the proteins had gene expression changes. Tubulin and microtubules, a heat shock protein, and some other enzyme I never heard of. The latter 2 having nothing to do with signaling or anything that could seem remotely applicable, and plus they're pretty small amounts. So it looked like proteomics and genomics pointed to microtubules as the site of action of anesthesia. And then they did another study in tadpoles, and they found this fluorescent anesthetic, an anthracene derivative, and it was only anesthetic when you fluoresced it. And they gave it to tadpoles who have conveniently transparent heads, so they could give it to them. They'd be swimming around, then they'd turn the UV light on, and the tadpoles would go belly up. They'd lose consciousness or awareness or whatever they have. I think they're conscious, actually, and go belly up. And when they sacrificed their brains, they found that the anthracene was bound to tubulin, to microtubules. And so this was consistent with quantum mobility, which was Hameroff's. This is Hameroff's lab, his name for our theory. It wasn't quite right, but it was good to get noticed. And this was optogenetics. And they also found clinical trials on patients on Taxol, which stabilized microtubules, required more anesthesia. So clinical trials, optogenetics, genomics, and proteomics all point to anesthetics acting on microtubules. Despite that, people still will say that they act on GABA receptors, even though that can't be true. So let's get back to the cell and look for. So here are the microtubules again. So why would binding of anesthesia to microtubules block consciousness? That's the question. So I've always thought that microtubules process information inside neurons, and here we see microtubules on the left underneath a synapse, the axon on the left. The dendrites where the microtubules are interrupted in a mixed polarity. And on the right, you see sequences of molecular automata simulations I did in the 80s and 90s. And I'll show you those references in a little bit. But the idea was that microtubules are polymers of the protein tubulin, which can be in at least two conformation or 2 states, which could interact by a local dipole-dipole couplings and process information kind of like cellular automata, or we call them microtubule automata, processing information. And it's likely that this could answer the question of how a single cell paramecium can learn, avoid predators, find food, find a mate, and have sex without any synapses. It's one cell, because everybody else would say, well, cognition and consciousness and all depends on network effects and through synapses. And the brain models that most people have is that each neuron is a dumb on-off switch. But yet we have these simple organisms like paramecium, which can swim, find food, and find a mate, and apparently have sex. They fuse and are absolutely motionless for about 30 seconds. So we don't know that they're conscious, but it could be. And also the slime mold, which I mentioned earlier, has now been shown to be able to solve the traveling salesman problem in these experiments where they put them in this well with food and a verse of things here and there. And it takes a while, but they're quite clever. And it looks like the microtubules are doing the information processing. So getting back to the paramecium, how does it do all these things? Well, it uses its microtubules. And upper left, you see a paramecium, and the little hair-like things sticking out are cilia. And upper right, you see a cilium in cross-section, which has 9 doublets of microtubules with a central pair. And lower right, you see a cross-section of the cilium coming out of the paramecium, where it can act as a sensor or as an oar to propel to move the thing around. And on the left, you see a centriole, which is a very similar structure of 9 microtubule triplets.
which are the core of the cell cytoskeleton and important for cell division and mitosis. So how do they bend? Well, atop you see a cilium, and then below it you see one microtubule with its dynein motor proteins, which with ATP contract, and they connect from one microtubule to the other. So when they contract in sequence, they cause a controlled bending, and they could either send a signal along the microtubule to the cell, the main part of the cell, or a signal could travel along the microtubule and cause these dyneins to contract sequentially, so you get a coordinated movement, and so that the microtubule was controlling the movement of the cilium. Well, it turns out the exact same thing is happening in our brains right now, even as we speak. This was done in 2008, and it has to do with synaptic plasticity. We see it below a neuron, and the axon is coming down to the lower left, and we see looking inside a dendrite. So materials for synapses that are to the right of the box, further along, will need to be upregulated or downregulated, and things are synthesized in the cell body and then transported along the microtubule by these kinesin and dynein, the exact same motor protein in the cilium of the paramecium, to carry the enzymes or the precursors or whatever is needed for a particular synapse. But how do they know where to go, where to get off? I mean, there's lots of synapses, and they have to jump from microtubule to microtubule, and they have to go left or right at bifurcations. Well, it turns out that tau, you can see there in the red, which is a microtubule-associated protein, encodes kind of a traffic signal for these motor proteins and tells the dynein and the kinesin where to get off and deliver its cargo. And so the placement of the tau on the microtubule lattice is essentially memory, because it's guiding synaptic plasticity. So this is more evidence that microtubules can process and store information. And of course, if the tau falls off, the microtubule destabilizes, or if the microtubule disassembles, the tau falls off. In either case, you get Alzheimer's disease. You get these neurofibrillary tangles of tau in the neurons, and then the microtubules disassemble. The neuron shrinks. You lose synapses. You get brain atrophy. Your brain shrinks. Now, despite this, and this was demonstrated in an excellent paper back in the 80s by Matsuyama and Jarvik, that microtubules were the key to Alzheimer's disease. Despite this, billions or trillions of dollars and not that useful research has gone into attacking the amyloid plaques in Alzheimer's, when we should be promoting the stabilization and the repolymerization of microtubules, in my opinion. And if we have time, I'd be happy to elaborate on that. It can be done with, I think, with transcranial ultrasound painlessly, safely, and harmlessly, along with a nice mood enhancement. But that's another story. So Alzheimer's and microtubules. So the idea that I was working on up until the late 80s, early 90s, was that microtubules were processing information. And I published a bunch of papers beginning in the 80s, and was going around to AI and neural net meetings saying that their target of brain equivalence was way naive, that each neuron wasn't a bit, that you had to go down to a deeper level. But before I get to that, let me say that there's evidence for quantum, not necessarily quantum, but for coherent dynamics in microtubules. And when Anirban Bandyopadhyay read about the work that Roger and I were doing, or even before even the classical microtubule work, he started studying microtubules, and in over 10 years came up with these studies at 3 different scales. The top, he's got nanoprobes in a little neural network where he can use 2 of them to stimulate and the other 2 others to record. And in the middle one, B, you see 1 microtubule between 2 X and Y electrodes, and then 8 other nanoprobes. And at the bottom, you see 4 nanoprobes around 2 rows of tubulins, just the proteins. So that's small, then medium, and then large at the top. And in all of these cases, he used 2 to stimulate. So if you just take a microtubule and stimulate it, it's an insulator. You're not going to get current. But if you stimulate it at an alternating current and sweep the AC frequency, you will find certain frequencies where all of a sudden the microtubule becomes highly conductive, ballistic conductance. It might even be quantum conductance, superconducting, if you could get around the interface between the probe and the tubulin. So that and it happened at every 3 orders of magnitude, every 3 frequency orders, and at each one he got a particular pattern called a triplet of triplets. So he would sweep the AC and get 3 peaks, and within each peak there were 3 peaks, and maybe if you look further you would see 3 peaks within those 3 peaks. I don't know, but you can see the 3 peaks on the right. You can see them in cross section, and he's called these octaves and various things, but they happen in terahertz, gigahertz, megahertz, and kilohertz, and even hertz. So this was very encouraging to Roger and me, and Honorbound's now a very good friend and collaborator on some other stuff. But this was very important. I think one of the most amazing studies done in recent years. It was actually a number of studies to put it all together. So anyway, that suggested that microtubules could process, say, in megahertz. And getting back to the brain computational capacity, what AI was saying in the late 80s, early 90s, and still saying, actually, the singularity, right, Kurzweil, that you have 10 to the 11th neurons in the brain, about 1,000 synapses per neuron, about 100 hertz, gives you 10 to the 16th operations per second. And Kurzweil and others were saying, well, when we get to 10 to the 16th, we'll have brain equivalence and consciousness. And I was going around saying, not so fast, because each neuron has about 1 billion tubulins. So if you look at microtubule computation, you have the same 10 to the 11th neurons, 1 billion tubulins oscillating at, let's say, 10 megahertz. They actually can oscillate at various frequency. That gives 10 to the 16th operations per second per neuron. So what they were saying for the whole brain, I was saying per neuron, and 10 to the 27th operations per second per brain. Well, AI people didn't like that too much. They didn't like me very much because, Roger was coming at him with his Gödel non-computability. It's hard not to like Roger, though. And I was being kind of a pest. And they said, go away, kid, you bother us. One day, though, somebody said, okay, wise guy, let's say you're right. And we have all this information processing. How will that explain consciousness? Because I was claiming it somehow gave rise to consciousness. And I had to admit they were right. I didn't know. They were basically saying, okay, where's the consciousness? Where's the being? Is something else required? Could it be quantum physics? And at least that's what Roger Penrose said, because this person who challenged me, and I'm very grateful, and I wish I could remember who it was, suggested I read a book by Roger Penrose called The Emperor's New Mind, because he had a mechanism for consciousness, which needed a quantum device, which I didn't appreciate at the time. So I read this book. It was an amazing book, mind-blowing in many, many regards, way over my head in most things, but with cute, very clever drawings and very cleverly written. And so I got a lot out of it. The first half was about Gödel's theorem and how consciousness couldn't be a computation. For understanding, for example, like a mathematician can't know a theorem is true. He has to be, a theorem can't prove itself. You need a mathematician outside the theorem to know that it's true. And knowing is kind of a feeling. So, and Roger was saying, he generalized to understanding is a feeling. And John Searle had said the same thing with his Chinese room argument. If you give somebody a Chinese symbol in a lookup table, they look it up and they can translate, but they don't understand Chinese. So for understanding, you needed something else, Roger was saying. And that something else came from quantum physics. And I didn't know very much about quantum physics, but I started to learn as much as I could. I'm still trying. So I'm going to give you my kind of amateur's approach to quantum physics, not being a physicist. And that is that the world is divided into 2 realms: the classical world, where everything is localized in one place, particle-like and fairly large, following Newton's laws, Maxwell's equations, et cetera, but at small scales, and we don't really know the cutoff, you can have quantum superposition where things can be in multiple states or locations at the same time.
Things are non-local, entanglement. They can be wave-like, and they tend to be small. But the boundary between them was a problem. And I've kind of come to believe that consciousness is actually on the edge between the quantum and classical. And in our case, Rogers and I, we think that the quantum gives rise to consciousness and the classical world. So we'd be going from the black to the gray to give consciousness. But other people would say that consciousness causes collapse, and you'd be going the other way. But in either case, you could think that consciousness is on the edge between the quantum and the classical. So, for example, let's start with the problem of superposition. A particle can exist as a wave of multiple possibilities shown in the background, or as a particle. This is a cesium atom, I think, in a definite state or location. Yet, when we observe or measure, we only see particles in definite states or locations. But before we even address that, Roger started off with addressing the question of how a particle can even be feasibly in a superposition of multiple locations simultaneously. And to do that, he accounted for superposition through Einstein's general relativity. And this showed that matter was equivalent to curvature in fundamental space-time geometry for large things like the sun, which would curve the starlight from stars behind the sun so they can be seen on Earth during an eclipse, which Eddington did in 1919 to prove Einstein's theory and win his own Nobel Prize. But Roger applied this to tiny things. What about matter and space-time curvature at small scales? So he introduced these two-dimensional space-time sheets with one dimension of space, one dimension of time, so you can draw it easily. And a particle would be a curvature in one location, evolving over time, and the same particle or different. It could move and have a different curvature. So an oscillating particle would be like two space-time curvatures oscillating back and forth. And a superposition then would be both curvatures coexisting. So you have a curvature, you have two curvatures, and essentially a separation in fundamental space-time geometry, so that the particle was temporarily at least in both. And this would be a separation in the fabric of the universe in space-time geometry. You can imagine that if these separations were to continue, you'd have multiple worlds. You know, the many worlds hypothesis, which, as absurd as it seems, is still, I think, the most popular view among physicists. Maybe Paige can comment on that. Then other ideas where the consciousness collapses the wave function. The consciousness shown here, Bing, and this was put forth by von Neumann, Wigner, back in the early days of quantum mechanics, Bohr to some extent, then more recently Stapp, David Chalmers, and Kelvin McQueen, and is often known as the Copenhagen interpretation after Bohr's Danish origin, which suggests that consciousness causes quantum state reduction, and I call this subjective reduction because the subjective person is causing it. And it causes one of the superpositions to cease and the other one continues. So it's causing collapse of the wave function. Now, how that happens is unknown. It doesn't explain what consciousness is. It doesn't explain what superposition is or how it could cause the collapse. But it's also dualist. It puts consciousness outside science. It doesn't even attempt to explain consciousness, and yet gives it some mystical, magical powers in collapsing the wave function. Roger said the superposition separation is unstable and will self-collapse, undergo objective reduction, OR, at time t equals h bar over E sub g. H bar is a Planck-Dirac constant. E sub g is the gravitational self-energy of pulling a particle from itself, pulling it apart from itself, so they're literally beside itself. And when this happened, there would be a moment of conscious experience. So he turned around the Copenhagen interpretation, which said that consciousness causes collapse, to say that collapse occurred spontaneously at time t equals h bar over E sub g, causing a moment of conscious experience. So rather than consciousness causing collapse caused or was equivalent, identical to consciousness. Now, these events occurring in the random environment, here, there, and everywhere, would be merely proto-conscious. They'd be random,
They wouldn't have any meaning, context, or memory. And I liken them to the notes, tones, and sound of musicians tuning their instruments before a symphony. It's kind of random, kind of a cacophony. It's noise, really. You could say proto-conscious noise here, there, and everywhere. Now that may seem strange, but panpsychists, of which many neuroscientists belong, claim that everything has some snippet of consciousness. So it's not that strange. It's a process, not a property. But how could they be organized or orchestrated to give rise to, metaphorically, music rather than noise? And so Roger needed a quantum computer in the brain which could biologically orchestrate quantum information, halt or terminate by Penrose OR, connecting to space-time geometry, and non-computable Platonic values and qualia, awareness and feelings, and could regulate functional neuronal and synaptic activities. So in his book, he didn't know. He put it out there. He knew that neuron firing and not firing was too big, was too macroscopic. But when I read his book, it hit me like a ton of bricks. He was looking for microtubules. He just didn't know about them. And so I wrote to him and suggested it, and the rest is history, more or less. And we teamed up to develop our theory of orchestrated objective reduction. So let's start with the tubulin molecule, which has, we were talking about these aromatic rings, tryptophan, phenylalanine, and tyrosine, and tubulin has a huge amount, 86 of them in every tubulin. And you can see them in the tubulin by color. And the red spheres are where anesthetics bind, and the anesthesia binds in and amongst the aromatic rings in tubulin. Okay, so what does that get us? So in developing our model, we wanted a something that would be macroscopic, involving the whole microtubule, and then multiple microtubules. So these aromatic rings can be aligned in various patterns around the microtubule helical lattice. And so just picking one to start with, we said that there could be a quantum channel going from, let's say, 8 o'clock to 2 o'clock through, and we just picked a few of the 86 to make it legible. And that this would wrap around the microtubule and could actually, the dipole oscillation, which are basically Frohlich oscillations, could also be, could oscillate between back and forth, but could also go into superposition of both orientations, and we have a qubit. And so this is the Orch OR qubit, the quantum bit for orchestrated objective reduction, which, by the way, also can give rise to topological quantum error correction, because you could have the quantum information going one way and the error correction going against the grain and intersecting with it and correcting as you go. Topological quantum error correction is possible. And as far as whether the propagation of quantum or the Frohlich oscillations are feasible, if you look at the just tryptophan, so here's a piece of a microtubule with 3 protofilaments, where we're just looking at the tryptophan, and you can see they're, at least along the vertical, they're fairly closely spaced, close enough to have quantum dipole coupling. And then side to side, because of the curvature, we think they may get closer that way. So it looks like we can have quantum dipole oscillations, or dipole oscillations, which could be quantum depending on other factors, in a microtubule. Now, just for your information, actin, and a lot of people say, well, why do you care so much about microtubules? Why not actin? Actin, I don't think, has the information processing capacity, the stability for memory, and also the tryptophans are too widely spaced. So it wouldn't be as good as microtubules for what we want them to do. So here we're back to microtubules, and we're back to honor bond, and we can see that we can have oscillations in terahertz, gigahertz, megahertz, kilohertz, and hertz in a microtubule. Going back to the Orch OR theory, when we reach the threshold, there's a bing. This is just for one piece of one microtubule, but we need a lot of microtubules, and I'll get back to that in a second. And then we have a series of conscious moments, bing, 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 and it gives rise to our stream of consciousness, and we go from there. And the rate can vary. When you're excited, you have more conscious moments per second, and the outside world might therefore slow down, and the opposite. And there can be a backward quantum information flow up to a point, which we can talk about later. So we wanted to quantify Orch OR, and originally we thought about decoherence or avoiding decoherence for 40 hertz for like 25 milliseconds, but that was unreasonable. And we decided that the Orch OR events must be much faster, like in, let's say, 10 megahertz. And then we calculated how many tubulins we'd need, and it turned out to be 10 to the 15th. And there's about 10 to the 20th tubulins in the brain. So this is about a 10,000th or 100,000th of the brain would be involved in one conscious moment, and they would have to be entangled. And I think that's possible through the what we call the quantum underground, even across neurons. And we have some evidence for that already. And the 10 megahertz will give you a favorably short decoherence time. So we can avoid decoherence for 10 megahertz for 10 to the minus 7 seconds. And then but that's too fast for cognitive epochs and gestalt scenes, and which lasts 300 to 500 milliseconds. So Roger came up with the idea that there could be interference among the very fast and beat frequencies, which could resonate and interfere across scale from terahertz to gigahertz to megahertz to kilohertz to hertz, and slower cognitive events. And in fact, in our 2014 paper, Roger and I proposed that EEG is actually coming from microtubules comprised of interference beats from faster microtubule dynamics. And 2024 is the 100-year anniversary of EEG, and we can now detect megahertz from the scalp in EEG, and we're preparing to say that EEG is actually coming from collective effects of microtubules, and that'll create a stir, and people won't like it. But I think even after all these years, there's not a better explanation. There's no unified theory of EEG meaning anything other than individual rhythms and bands. So we'll see how that goes. Anyway, this led us to come up with this scale-invariant hierarchy, in which we think consciousness is happening. Now, what you see here on the left is a pyramidal cell neuron. And I think for reasons I don't have time to discuss, they are the most likely site for consciousness. And the microtubules and the dendrites and soma are broken and interrupted on a mixed polarity. So they're going to be slightly different energy and will give rise to beat frequencies. Anyway, the frequency for the neuron is in the hertz, like EEG. As we move to the right into microtubule networks, we get 1,000 hertz. Going further, we get a megahertz, a million hertz, and then a billion hertz for rows of tubulin dipoles, terahertz for tubulin, and then petahertz for pi resonance dipoles, and then 10 to the 18th for the atomic nuclei. And I forget what you call 10 to the 18th. And then big step from 10 to the 18th to 10 to the 43 hertz to the Planck scale, to Roger's objective reduction. And so we're basically saying that there's a hierarchy that extends. Now, a lot of people talk about hierarchies from neurons going upward in size, getting slower. So like kind of EEG type rhythms going upwards. But we're saying, look inside, go inside the neuron, and we have this 12 orders of magnitude in frequency going down, including getting into the quantum realm. And actually, Anirban calls his 12 order of magnitude EEG device the dodecagram, the DDG, for 12 orders of magnitude. As I said, we're messing around. We have one probe here in Tucson. We're looking at megahertz. We hope to put an animal to sleep next week and see if it goes away. That's completely harmless, I should add. I've done it to myself many times. So anyway, we have this multi-scale hierarchy, and I think consciousness can happen at various places in this hierarchy, or a combination of places, kind of like music. This is kind of like the quantum orchestra, I like to call it. And I think if you look at EEG only, or just measure in hertz, you're only like going to the symphony, you're only hearing the low bass. You're not hearing the rest of, you're actually not hearing the music. You're just hearing one aspect of it. And I think if we go inside and get into the kilohertz, megahertz, gigahertz, and terahertz, we'll find a lot more richness and get closer to consciousness, which can happen, I think, at any transition between these frequencies.
And I say that because of this work by Greg Scholes, who was a collaborator on the paper I'll tell you about in a second. Is a well-known quantum biologist at Princeton. And he was looking at an aromatic system there, and which was oscillating in petahertz, which is where we think the dipole oscillations are happening. And there, he noticed that there's an abrupt loss of phase coherence along the high frequency petahertz vibrations, followed by impulsive appearance of phase coherence along lower frequency terahertz picosecond vibrations. The coherence jumps down 3 orders of magnitude as a quantum beat, and new wave packet appears at this lower frequency. I wonder whether this is actually a collapse of the wave function going from, say, petahertz to terahertz, or even further down. And I'll have to check with Roger on that. But this is the beat side data is exactly what we think is happening. And this whole thing may be more like a multi-frequency scale invariant hierarchy, as I said before. How are we doing for time? I'm over already, huh?
It's well,
Okay.
if you're available, I'm not going to stop you, and we'll be recording for those that have to leave.
Okay, I'll keep going then. Am I okay?
Yeah, please.
We did this study. Let me go back. We did this study, Travis Craddock, Jack Jasinski, some others, looking at tubulin. I mentioned these aromatic amino acids. Basically, they modeled, so I showed you before the oscillations between two benzene rings in the terahertz. They modeled all 86 of these aromatic rings oscillating with all the other ones. And it was a super duper computer somewhere back east, I think at Johns Hopkins and Philip Currien's lab. And we found at ambient temperature a spectrum of collective dipole oscillations with a common mode peak at 613 terahertz in the blue light region of the spectrum. And anesthetics and also non-anesthetics had their own frequency emissions. So up here, TFMB, I'll tell you about in a second, is a non-anesthetic. F6 is a non-anesthetic. Meth is not like the Breaking Bad meth. It's methoxyfluorine, a very good anesthetic. Death is not death, it's diethyl ether. And this is desflurane. So each of these, anything that's not pointed to by one of these is actually coming from the tubulin itself. So all these aromatic rings are oscillating and giving the spectrum of in the terahertz. And when you put the anesthetic in, let me back up and say that there was a common mode peak from all these at about 613 terahertz. But when you added the anesthetic, it abolished the 613 terahertz peak and dampened the other frequencies proportional to the anesthetic potency. So this is kind of like a Meyer-Overton correlation for on a computer simulation of terahertz oscillation. So we did subtraction spectrum for all the anesthetics. And if you see a negative here, it means that it had disappeared from the original spectrum. So it disappeared for desflurane, for diethyl ether, enflurane, halothane, isoflurane, methoxyflurane, nitrous oxide, sevoflurane. But then there are these other anesthetics, these other molecules that are not actually anesthetics. So I'll come back to them in a second. The point of the main study is the presence of anesthetics abolished and non-anesthetics did not abolish the 613 terahertz blue-green peak. So I don't know the significance of that peak, but it does go away with anesthesia. Now, there are several gases, and so that's a unitary hypothesis, and somehow that frequency must be necessary, or some frequency must be necessary for consciousness. Now, let me just digress a second. There are non-anesthetics discovered by Eager's group years ago that follow Meyer-Overton and bind in the same places that the anesthetics act, but do not cause anesthesia or loss of consciousness. They're called non-anesthetics. Trifluoromethylbenzene and F6. I forgot what that stands for, but these are hydrophobic nonpolar molecules, which do follow Meyer-Overton, but don't cause anesthesia. And they were discovered by Eager's group back in the 90s. They disobey the Meyer-Overton hypothesis because they bind where they should, but they don't cause anesthesia. Why not? Well, first of all, here's F6 and TFMP, and we've got them at a MAC of 1,000 or infinite because they don't cause anesthesia. But if you look at where they are on the oil gas partition coefficient, they're right in there. They're between methoxyfluorane and halothane, and right about where sevoflurane is, which is the anesthetic I use on a daily basis. But they don't cause anesthesia. Why not? Well, Travis came up with this, I think, brilliant idea. Well, first of all, here's to show that the F6, it doesn't reduce the peak at 613. TFMP actually increases it. And fluorothil, which is a non-anesthetic and also a convulsant, had a negative peak. So these things did not dampen or inhibit the 613 peak. Travis then modeled the polarizability of all the anesthetics and the non-anesthetics. And this is very interesting. He found that all the anesthetics, nitrous oxide, desflurane, diethyl ether, et cetera, fall pretty much along the straight line of MAC. So MAC correlated with their polarizability. The more polarizable, the lower the MAC, the more potent. So methoxyfluorane is the most potent. Nitrous oxide is the weakest. That's very interesting. However, F6, which is not an anesthetic, and TFMB, which is not an anesthetic, their polarizability was greater than methoxyfluorane. And we postulate that the polarizability of these anesthetics is too great. And therefore, they don't cause anesthesia. They don't disperse the dipole like the anesthetics do, the van der Waals dipole dispersion forces, but they just go along for the ride. So here's the non-anesthetic, and here's the dipole oscillation. It's just going to oscillate right along with because it's highly polarizable. It's going to go along for the ride with the intrinsic dipoles. But the anesthetic has some, I don't know what to call it, drag or something. It's not polarizable, so it doesn't go along for the ride, and it messes up the dipole oscillations of the intrinsic aromatic rings. So we think we solved that issue, that the non-anesthetics aren't anesthetic because they're highly polarizable and quite literally go along for the ride. Okay, I'm going to finish up here, and I apologize for going over, but I think this is kind of interesting. When did consciousness arise? And here's the history of the big universe since the Big Bang. This was done around 2000. And most people would, no, not most, but many people would say that fairly recently with tools and language. Other people would say the Cambrian evolutionary explosion, which there was a huge burst of activity around 540 million years ago, where all the animal phyla appeared. And we know from fossil records that these organisms had these spiny things. This is very much like the actinospherium. This is like a modern a suctorium, and that's a troglodyte or something, I forget. But we know this is like the actinospherium that John Nunn studied. And so we think it has microtubules, and the suctorian is still around, and it has this sucking thing that is made of microtubules which interact to contract, so it can literally suck. And so microtubules go way back to the Cambrian evolutionary explosion, it looks like. What about animal cells? Well, animal cell eukaryotes, they came along when, according to Margulis Sagan, when mitochondria entered the prokaryote and when spirochetes invaded the prokaryote, and kind of like the sperm and the egg, and the spirochete gave the cytoskeleton and allowed mobility, motility, and intercellular mobility as well. So that would be the first animal cells. And here's the sperm entering the egg, like the spirochete might have entered a prokaryote millions of years ago. And here's the
possible information in the flagella of that spirochete that could convey epigenetic information, for example, to the egg or the prokaryote. It's also possible that consciousness began around when life began, or even before. Colin McGinn has suggested before, and Roger Penrose has this idea of serial eons prior to the Big Bang, and there were presumably consciousness in a previous eon. But let's back up, because we should ask, which came first, consciousness or life? And there are two, most people would say neuroscience. In neuroscience and Western philosophy, people would say life came first and preceded consciousness, and consciousness emerged from life. And that's what most people think. However, panpsychists, Eastern philosophers, spiritual people, Whitehead, people who follow Penrose objective reduction, including me, would say that consciousness preceded life and that life emerged from consciousness. Now, that seems to get it backwards, but I think it may be right. Think about this. Life on Earth apparently began in a primordial soup proposed in the 1920s by Operin and Haldane, a shimmering, oily mix from which biomolecules emerged 3 to 4 billion years ago. And in the 1950s, Miller and Urey simulated a primordial soup with the constituents that they knew were there, and sparks for lightning, and came up with something in this flask. And when they looked, they found amphipathic biomolecules, organic rings with polar water-soluble tails, much like aromatic amino acids or lipid molecules, or even nucleic acids. And these would be attracted by their transient-induced dipoles to form this micelle, according to Operin and Haldane. And here's a more realistic depiction by Arvydas Tumulus in Lithuania of what the aromatic rings coalescing in a protein might look like, because you need a fair amount of them. But the idea, and I proposed this in a paper in 2017 on the quantum origin of life, did the brain evolve to feel good? That back in the primordial soup, that there reached critical threshold for a proto-conscious moment at time t equals h bar over e sub g, and there was some kind of proto-conscious moment in these primitive micelles. Now, these would generally tend to be random, but occasionally some would be positive and feel good. I think I'm in the way of a smiley face there, but you can imagine, I'll do my own smiley face. You can imagine that some of these would feel good. And this could be this pleasure could be a feedback function orienting pi resonant groups to optimize pleasure. So did life then evolve to orchestrate and optimize objective reduction, OR mediated pleasure? And I call this the quantum pleasure principle, after Freud, who had, of course, the pleasure principle in his book Beyond the Pleasure Principle. So I guess quantum is beyond. So I think, Darwinian evolution is a pillar of neuroscience, and you can't really criticize Darwin, who wrote a lot about consciousness and mind, but didn't include it. But the notion that life evolved behavior to promote gene survival, which I guess is Dawkins and others' idea, is an assumption. It doesn't really make any sense. If a gene doesn't feel, why would it? What's its motivation? Behavior is driven by reward from every rat in a maze in a lab, pleasure avoiding pains. There was no genes in the primordial soup. And unfortunately, evolutionary theory ignores consciousness and feelings because it doesn't have a mechanism. In fact, other than high order emergence, which is kind of a hand-waving argument, I think Penrose OR is the only actual scientific proposal for conscious feelings, for qualia. So did feelings drive evolution? I think so. I think it's a radical idea, but I think it makes a lot of sense. If we have a mechanism for consciousness that was there, then all that behavior from first life to even when you get to genes millions of years later, how did that happen? This would explain it. And this is also called the phenomenal argument in evolution. And just speculating a little bit further, it could be as simple as when the aromatic rings collapse, they can be in a couple different conformation that are stable, the T and the parallel offset, and maybe these correlate with simple proto-conscious feelings. And we could even have a qubit of a superposition of of both states collapsing to one or the other. Now, it's probably not that simple. On the other hand, we don't know. And if those two aromatic rings can give that, when you have 86 of them, and believing that the emojis can represent qualia in some way, we could have quite a bit of a range of qualia, even in one tubulin, that could entangle and couple and correlate with all the others. So back in the primordial soup, anywhere else, yes, aromatics are everywhere in the universe. And here are some aromatics, buckyballs, nanotubes, and polyaromatics floating in the universe. And they're generated by stars. And they're called polyaromatic hydrocarbons. They're prevalent in the universe. They're in interstellar dust, atmospheres of, and they apparently even float to Earth. They were found on the windshield of the International Space Station. My friend, University of Arizona planetary scientist Dante Loretta, headed NASA's OSIRIS-REx project, which after 7 years, just a couple of weeks ago, brought back samples from the asteroid Bennu. And Dante hints to me that he got lots of polyaromatics, and he'll be announcing that soon at a big conference. And we intend to study these with the same type of studies that we do to test for consciousness, like to look for quantum optical effects, to see what aromatics or even psychedelics might do, or to see if these molecules might have pharmacological effects. This is a molecule that came from a meteorite. That's quite a complex molecule. And Anirban says that if you let this find its shape, it could be self-replicating. So that's something else we're gonna look for. Much like he's working on his self-organizing quantum warm temperature quantum computing. And they come in all shapes and sizes, and maybe they're trying to tell us something. And in this picture, the green from the interstellar dust is fluorescence triggered by starlight from PAHs, polyaromatic hydrocarbons, in ice in interstellar dust. And we're being radiated by terahertz from these even as we speak, assuming it gets through the atmosphere and stuff. So let me wrap up and just mention the Penrose-Hameroff Orch OR theory, and claim that it has more explanatory power, connection to biology, and experimental validation than all the mainstream theories, which are largely what I call cartoon neuron theories, because they just consider neurons as empty and a 1 or a 0, and don't consider and only operating in hertz. So like I said, if you only consider that, you're just hearing the bass sounds at the symphony. You're not really hearing the music, if what I'm saying is correct. I should also say that just based on the number of tubulins, we can say that all organisms, including plants, have consciousness, but at a different frequency. We might have 10 to the 10th tubulins, 10 to the 10th conscious Orch OR conscious moments per second, which interfere down to give us gigahertz, megahertz, all of them, all those frequency ranges are down. But a mouse only maybe 10 to the 6th, a worm maybe 10 to the 3rd, a paramecium maybe 10, and a plant less than 10, or actually one every conscious moment every couple of minutes, something like that. So it's just a matter of the frequency of the conscious Orch OR event. So in conclusion, thanks for your patience. Let me say that life may be quantum in nature, its essence in decoherence-free subspace, the essence of life in decoherence-free subspaces in what I'm calling the Meyer-Overton quantum underground, and that regions of aromatic rings and other nonpolar biomolecules sequester, avoid water, and avoid polar charges of all kinds, enabling a limited degree of freedom and quantum mechanisms with limited degrees of freedom to be isolated adiabatically without heat exchanges in their solubility compartment, assuming they're arrayed in lattices so they can couple and entangle periodically to give coherence, as we seem to see in microtubules. And consciousness in the brain is likely to occur in a scale-invariant hierarchy in mixed polarity microtubules in cortical layer 5 pyramidal dendrites and soma, where the microtubules are mixed polarity,
again, more like a quantum orchestra than a computer. And I didn't really talk about the brain very much, but this fits very nicely in the pyramidal cells. And finally, the proto-consciousness, I think likely preceded life, prompting its origin and driving evolution. And I want to say that a lot of these ideas are based on Roger's ideas and Anirban's ideas, and my other colleagues, Travis Craddock, Jack Tusinski, Travis Craddock, and Arad Kalra, and others. So with that, I'll end, and thank you for your attention.
Thank you so much, Stuart. That was fantastic. It really filled in a lot of details of that I haven't seen in your previous talks before on your videos. We've got some hands raised already.
I can't, so please call on for me.
Derek, Jaroslav.
Yeah, Yarek is fine. Stuart, do you hear me all right?
Yes, I do. Thank you.
Okay, so sorry I came late to your seminar, which I regret because it was excellent. But the general feeling I have from your talk is the dynamics plays much bigger role than the static structure, right? Statics, of course, organization of the molecules, but the dynamics, how they bend, how the phononic modes are going through them, what you call plasticity. Is it a right assumption? If this is a right assumption, then that 10 to the 27 operations per second needs to be multiplied by all those different dynamical modes, right?
I'd settle for 10 to the 27th, but you may be right. I mean, yeah, if you want to go down even into Planck scale geometry, yeah, it'd be vast. But let me back up to your first point. I agree that the dynamics, and particularly oscillation, I think when we first started studying microtubules, we were looking for something propagating along, carrying a signal. And we did see these excitons propagating along. Oh, I didn't show that. But we, as part of the Templeton, we did tryptophan fluorescence lifetime, and showed that if you hit a microtubule with UV and put a label further down, that the label will emit because the exciton is propagating a few tubulins and then emitting further. And then if you give anesthesia, you inhibit that. So we were looking for something propagating, but Anirban has convinced me that that's not really necessary. You want oscillation, and then you can get signaling and information transfer by phase differences, by phase interference and so forth. And so the system's probably way more sophisticated than we can even imagine at this point. But I think the frequency oscillation, that where you have the self-similar triplets of triplets from terahertz, gigahertz, megahertz, kilohertz, and probably hertz, is the key. And that's something that we just need to look at more carefully. So I don't think we're sending dynamics doesn't mean sending an impulse down a microtubule like an action potential. It's more oscillation and phase interactions which give rise to information.
Thank you.
You're welcome.
John Holler, go ahead.
Yeah, so I put my questions in the chat there, but I have sort of two related questions, sort of related. One was obviously this was a great lecture, by the way. It was fascinating, but obviously it's rather controversial, as you pointed out in the beginning. So I'm wondering, how this OR theory, which posits conscious modulation through these microtubules, microtubules can be experimentally verified or disproved. So what are sort of the critical experiments? And then I have a maybe it's an unrelated question. Can consciousness be measured with a squid or some modified magnetometer?
Well, the last question, my friend Hartman Nevin is trying to do something like that at Google. He's the head of quantum AI. So I'm not sure, but in terms of your previous question, how can Orch OR be tested? We were in the Templeton World Charity Foundation Accelerating Research, what's it called? Accelerating Research and Consciousness Program, where they funded adversarial collaborations, where they started with 5 theories, IIT, global neuronal workspace, higher order theory, predictive coding, and Orch OR, and invited us to meet with the others and come up with experiments that one experiment between 2, which if it went one way would falsify one, if it went the other way would falsify the other. So we met with Giulio Tononi and Christoph Koch and others, and Roger was there and Honorbaum was there, and we couldn't come up with a common experiment. I don't think IIT is testable at all, actually. And what we proposed was to, 1, demonstrate a quantum effect in microtubule at warm temperature, and then 2, show that it's inhibited by anesthesia. And we wanted to, and then Templeton, the adversarial collaboration fell through, but Templeton gave us $200,000 to do the experiments. And we did the experiment at Princeton in Greg Scholl's lab by Ara Kalra. And it's what I mentioned this before, that he hit the microtubule with UV. You got a signal, exciton propagated several tubulins down, emitted a different wavelength from, so we knew that the exciton or something propagated. And then we gave anesthesia and it inhibited that significantly. So when I proposed that could falsify Orch OR, a lot of my colleagues said, whoa, you went way too far, because it needn't. And then even Roger said, well, it could be some other mechanism for quantum, not involving the quantum optical effects. And he said, well, you've put all your eggs in one basket and my eggs too. So I felt a little bad about that I'd gone out on that limb. But fortunately, I felt it had to be that way for a number of reasons. And I had published that in a paper on surveying theories of consciousness. And fortunately, the experiment worked out. But unfortunately, I don't know if you followed all the hubbub with the IIT and GNW showdown, and then all the people calling IIT pseudoscience and the big bet at ASSC. And so nobody talked about, so all those theories fail to show evidence for themselves or disprove any of the others, whereas our experiment called its shot. I said, we'll show a quantum effect at room temperature. That was considered unlikely. And then they would go away with anesthesia. Well, both of those worked. So I was feeling pretty good about it, but nobody noticed because, all the big shots, Christoph Koch and Chalmers and Tononi and they, all the journals wrote up about this stupid bet. And I told Christoph, I said, well, quantum effects in the bet was made 25 years ago at our conference, actually, whether there would be a neural correlate of consciousness in 25 years. So 25 years later, they called the bet, and Christoph had to admit that there was no neural correlate of consciousness. So Chalmers won the bet and got a case of wine. But I told Christoph, I said, well, quantum effects in microtubules are a good neural correlate of consciousness. You could have won the bet. And he basically said, well, blah, blah, blah. He wouldn't want to admit it. He's been against this idea all along. So that experiment is already done. And that's more conclusive evidence than any other theory has for any other.
So that, if I can just quickly follow up, that demonstrates that you can disrupt quantum phenomena in microtubules, but I don't see the connection to consciousness. I think your theory says that anesthesia equals loss of consciousness. Therefore, if anesthetics disrupt or are correlated with these quantum phenomena in microtubules, those things are all equivalent, but we all know correlation doesn't prove cause and effect, right?
That's true, but anesthetics take away consciousness, and we made the prediction, and it came true. I guess what you're saying is we should try non-anesthetics and see if they do the same thing. And we plan to do that, and we plan to do other things like looking at measuring a rat's megahertz, which we know comes from microtubules, giving it anesthesia and seeing if the megahertz goes away. That would be another. It wouldn't be conclusive because you could make up some other story. But if you make a prediction and it comes true,
Right.
that adds evidence. It's not
Right.
conclusive. And we also want to look, honor bonds show an entanglement between microtubules. We want to see if anesthesia takes away entanglement between microtubules, takes away the terahertz, gigahertz, megahertz, and see what it does in his organic brain jelly. See if that may be conscious. I don't know. So I would say that we have, in terms of supportive evidence for our theory, we certainly don't have any conclusive evidence, but we have 1,000,000 times more evidence than any other theory, which is 0, I would say. Because I mean, their predictions, well, if we do this and that, we'll get MRI activity in this part of the brain. Well, so what? You don't even know how MRI activity correlates with consciousness. It's blood flow. And EEG is just probably coming from microtubules anyway. So we have evidence, they don't. I'll just stick with that for now.
Good, thanks.
Okay, Sita?
Yeah, thanks, Stuart. Excellent presentation, great history of the anesthetics. And essentially, you are starting a quantum pharmacology of consciousness. That's kind of the way you are going here. But one of the things that really intrigued me about your microtubule theory is that if you look at the cellular compartments with so many different organelles, is microtubules really controlling all these conscious and all these biological effects? What about organelle like mitochondria, which controls the energetics of the cell? And there has to be consciousness and movement and feelings, and all of that has to be related. So how is that only microtubules seems to be controlling this? Am I missing something here?
Well, I think mitochondria are important. They make ATP and they give off photons, and the photons may be useful, maybe relevant. I don't know. But basically, they're the power supply. We got I guess the liver would be our power supply. It takes food and makes stuff that we need. The heart is a power supply, pumps stuff around. But the brain is where consciousness is. And you could say, okay, well, maybe there could be consciousness elsewhere in microtubules, and that's kind of another story. But I think the microtubules are basically the nervous system of the cell. And the centriole in particular, which I didn't talk about, would be more like the brain. It's the central focus of the cytoskeleton. And the centriole has one of the cilia sticks out through the membrane and kind of acts like an antenna sticking out into the neuropill. So I would say that, anything with a hydrophobic pocket in a cell could entangle and couple to the microtubule oscillation, kind of like the rhythm. And so other things could be involved, like a GABA receptor on the membrane. It binds anesthetics, so it has an aromatic center. And those, when the microtubules are oscillating, that may be oscillating right along with it. And a lot of things, actually, anything with aromatic rings might get coupled into the synchrony. So it could all be synchronous. But I think, like I said, that the microtubules and the centrioles particularly are the brain and nervous system of the cell.
Yeah. But I'm really fascinated because there has to be a connection between microtubules and how the mitochondria provide the energy, all the fast electron transfer mechanisms through which it produces ATP and contributes to the motion. So that's why I'm thinking that there is a connection between microtubules and the mitochondria working together, if not for other organelles in the body. I may be wrong about it, but I think there is a connection.
You could be, and you could be right. Actually, the mitochondria are very closely associated physically with microtubules. They get moved around. They actually sit on a microtubule and get not like those motor proteins because they're too big. And so they get transported around by microtubules, and they could be getting energy directly from. But overall, and a lot of my friends, push mitochondria. There's a whole clique of people on Twitter that are pushing mitochondria. And I just think that they're important for energy. Who knows, they came endosymbiotically also. They could have something going on their own. But again, microtubules seem the most likely to be information processing devices and the thing that's organizing stuff inside the cell.
Okay, thank you. Thank you. Fascinating.
You're welcome.
Paul, go ahead.
Yes, I'm very interested in the qualities of microtubules that might allow them to be effective quantum sensors. So you had mentioned that a Taxol experiment, which selectively affects depolymerization. And in neurons, most microtubules are actually quite stable and are not affected by Taxol. So are there experiments that might help identify what element of the microtubule system and in what state of assembly and disassembly are critical for quantum sensing?
Yeah, it's not so much disassembly. As you say, the microtubules in neurons are stable because neurons don't divide. So they don't have to all of a sudden disassemble and make mitotic spindles for mitosis and then go back to doing what they were doing before. They just stay what they are. In dendrites in particular, they're capped at both ends. So they don't treadmill. Some microtubules, they'll lose tubulins from one and add from the other. They call it treadmilling. But if you did that, you'd be replacing the, well, let me back up. I think memory is stored in.
It would be more dynamic instability from the plus end rather than treadmilling from the plus and minus end in neurons as well as most other systems.
In dendrites, they're capped though, and they're capped so they don't do that. So the lattice configuration stays the same. So it's a perfect memory system because it's kind of like a mosaic. Each tubulin can be actually 22 isozymes in the brain, post-translational modifications. So you can have like 30 different types of tubulin in any one microtubule. So you have a mosaic. And that's going to affect its vibration. So I think it's a perfect memory. In fact, I saw somebody asking about memory. We have a paper. So everybody says, okay, memory is in synaptic plasticity, but the proteins in the synapses last hours to days, and memories can last lifetimes. So how's that happen? So, and the synaptic plasticity is controlled by microtubules. The microtubules, again, they don't disassemble for division. They're capped at both ends. So we think that we have a paper from 2017. Travis is the first author, Travis Craddock, where the calcium comes in, activates this enzyme, CAMK2, calcium calmodulin kinase 2, which transforms into like, it has 6 kinase domains in a hexagon above and 6 kinase domains above and below this holoenzyme. And when I first saw that, I said, a hexagon, that looks about the size of the tubulin lattice. And so I asked Travis, and he did the modeling, and he found, and it turns out it matches perfectly. And we think that CAMK2 phosphorylate, each of these 6 kinase domains can phosphorylate a tubulin, and you can phosphorylate 6 tubulins at a time. So it's like 6 bits of information with each CAMK2, and you get like hundreds per neuron and over thousands of neurons in an area. So we think that's how memory is encoded in the microtubule from the synapse, and then the microtubules control the synapse. So I think memory is in microtubules.
Right. Yeah, and I'm suggesting more experiments on the electrical and biochemical properties of the microtubules would be a really fruitful thing to follow.
I'd love to do that. You know, we want to, we want to do more. And so if you have any specific ideas, let me know. You know, we're doing Honorbond, we're going to be doing some experiments. Well, one thing I want to do is, for example, we did an anesthesia experiment and showed that it inhibited, but what about psychedelics? I mentioned that the 5-HT2A receptors are inside neurons too. And I suspect that these small molecules get in and don't even need the 5-HT2A receptors and could just bind directly to microtubules. And I would predict that they would increase the frequency of oscillations and tend to push the microtubule more into the quantum realm. As you go faster and faster, you get more quantum. So psychedelics are, was it you mentioned quantum pharmacology? I think that's exactly right. You know, I've talked to a lot of pharmacologists over the years, and I asked them about the aromatic rings, and they said, oh, we hate those. They're grease spots, or they mess up our gels or something. So I thought they were ignoring the most interesting part. So if you have any specific experiments, please email me.
Great, thank you.
Welcome.
Enjoy the talk.
So Stuart, just building off of the your comments about the CAMK2 and storing memory. So am I interpreting it right that phosphorylated or not phosphorylated would translate to like a 1 or a 0 in storing information. Is that sort of the model that you're talking
It
about?
could be. It could be. I think it could be that it just kind of energizes that system. It also brings up the idea of spin, of nuclear spin. I didn't have time to talk about this, but xenon. There's a study a few years ago in anesthesiology from China, and xenon is an anesthetic, and it's also an atom, so it can have isotopes. So they found different isotopes of xenon, one of which had spin 1/2. Regular xenon doesn't. And they gave it to mice, and they measured MAC, the writing reflex, and they found that the xenon with the spin 1/2, nuclear spin 1/2, was significantly weaker anesthetic than regular xenon. And they concluded or asserted that this was because the spin was promoting consciousness. It was making the animal more conscious, and therefore kind of antagonizing its own anesthetic action. And anyway, anesthesia, they submitted to Anesthesiology, which is #1 journal in our field, and Anesthesiology didn't know what to do with it. They sent it to me to review, and I loved it. I thought it was a really, really cool paper, and I wrote a very positive review of it. And so they asked me to write an editorial, which I did. And basically, and I asked Roger and some other people about the spin, and he said, well, spin will promote entanglement. And we know from these, what do you call it, paired radical pairs, that nuclear spin can even affect electron spins, I think. So it could be that having more nuclear spin could promote consciousness. That's what this experiment showed. So going back to the phosphorylation, I think phosphorus can have a spin too. So maybe that plays a role. But I kind of think that just putting that phosphorylation energy is just kind of it could add more. It does probably add more memory, but or more information, but could also kind of activate that particular memory to become more into consciousness for whatever reason.
Okay, and then just curious that, like, could those tubulins that are storing that memory, could they still become part of a qubit, or are they like sort of
Yeah.
Okay.
Yeah, again, our qubit, the way I think of it, is kind of a helical
Right.
dipole, and they'd be, somewhere in there, and that also gives you decoherence, topological error correction, because if one of them gets knocked out of whack, the others will pull it back into coherence. So, but yeah, it could still be part of a qubit, absolutely.
Okay, and then just looking at some of in the chat, if consciousness is a computation of microtubules, then what are the synapses for?
Slow stuff.
Okay.
I hesitate to say it's a computation. Roger says it's not really a quantum computation. It's kind of like a quantum computation until collapse, and then it collapses by OR. But it's like a computation. But the synapses would be for slower stuff. So imagine you had a single cell, and it got in cahoots with another cell and wanted to communicate, something like that. So.
Okay, and I had a question about the wave function collapse, the spontaneous collapse, whereas like you mentioned, most people have the belief that it's observation or consciousness that makes it collapse. And I was wondering if those two theories are at odds. Is it that it will spontaneously collapse if it's not observed, after time t, or is it that every time it collapses, it's not because of observation? Do you know what? I'm having trouble forming the question, but.
Well, yeah, it's Dave Chalmers and Kelvin McQueen have taken up that idea that consciousness causes collapse, going back to von Neumann, Wigner, Stapp. And again, it puts consciousness outside of, it's dualist. It puts consciousness outside of science. And Chalmers is okay with being a dualist. And they try, I said, well, where does this consciousness come from? And they try to go into the multiple worlds thing, and I had to bring up the anthropic principle, and that stopped that argument. But it's kind of like they're coming from, we're converging on the wave function. They're saying consciousness causes collapse, and we're saying collapse causes consciousness. And Dave asked me, well, are you saying collapse causes consciousness or collapse is consciousness? And I can tell a trick question a mile away. I knew this was a trick question. And I said, okay, I'll bite us. And to cut to the chase, he said, if you say it's causing, collapse is causing consciousness, well, there's a lot of things that cause consciousness. Breathing causes consciousness, or is required, or you know, metabolism, or this or that is all causing consciousness indirectly. And I said, okay, well, what if you say it's I said it's an identity. He says, well, identity theories don't work. I'm going, why not? And he cited the neuron, Pat Churchin's neural identity theory, that neural activity is consciousness. I said, well, that doesn't work because that's stupid, but this does work. So ever since that I've stuck with the notion that of identity theory, that consciousness is collapse is consciousness. And that means that everywhere it's happening at the ultra micro level with these proto-conscious blips, and we don't even notice.
I guess I know there's a lot of people with questions. I don't want to take up all the time, but just to clarify, so take like the double slit experiment, you know, where basically measuring where the electron or where the photon lands forces it to pick a spot, right?
Right, that's a good one. And so as you know, you send a wave, it goes through a slit. If you observe before it collapses and you see a particle pattern, you see only the slit. If you observe it afterward, if you observe it in the waveform, it collapses. So how does conscious observation of that cause collapse when it's in the waveform, if it's a wave? And I think the answer is that collapse happens at time t equals h over e sub g. So you have the e sub g of the particle or the electron or the photon, whatever it is, and you have the e sub g in your head of the consciousness. And when you entangle, you observe. And at some point in this process, the e sub g in your head, which hasn't quite reached threshold yet, is going to summate with the e sub g in the particle, in the device, and it's going to hit threshold, and that's going to collapse. So I could see consciousness causing collapse in that way, involving entanglement. But that's not what Chalmers and those guys say. They don't go there. They don't have a mechanism, but because they won't go for e sub g, they won't go for the t equals h bar of e sub g. But if you use that equation, then you can just say, well, if you add your consciousness just before it reaches threshold and the consciousness of the particle, you reach threshold, it collapses, you have a particle, and you have the experience of seeing a particle or observing a particle.
Okay, that makes sense.
It's different from Schrodinger's cat because you're not bringing, it's kind of like, well, I don't know if it's like, it's not like Schrodinger's cat. It's trickier than Schrodinger's cat. Schrodinger's cat, I would just say, well, if the cat could be in superposition within like 10 to the minus 30th, a 1 kilogram cat within 10 to the minus 30th seconds, it's going to collapse one or the other. So you never know the difference. I think the real question there is the live cat is a quantum entity too. I think life is a quantum entity.
Okay. I'll have to sit with that for a little bit. Okay, so there's some more hands up. Alistair, please go ahead.
Hi, Stuart. Thank you very much. Been reading your work for ages.
Thank you.
Yeah, very, very interesting. A couple of questions and a comment. The first question, of course, is you're suggesting the role of magnetic fields and spin, and this has been quite well shown now in controlling metabolism. Of course, this suggests something about consciousness being tied into the magnetic field of the Earth. So what happens if we leave the Earth and go outside of the magnetosphere, which would be quite interesting, which is something which hasn't been explored in any detail yet, but we certainly know when astronauts go into space and we look at some of these experiments where we take away the magnetic field, we're getting quite big metabolic changes. We've been interested in this certainly from the mitochondrial perspective, and certainly a comment made earlier. Mitochondria, of course, much more integrated because, I mean, for instance, tubulin modulates mitochondrial function, say through VDAC, which is quite well known. But what would you say about the role of gravity in consciousness as we go into, if we go into, say, zero gravity fields, well away from a big planetary body?
Yeah, well, the first question is what detects gravity? And again, the answer is microtubules. I think there were studies back 20, 30 years ago of putting these cell cultures in space, or just putting them in a thing on your benchtop and rotating them. So the average gravity over time is zero for any one of them. And they determined that it was the centrioles and the centrioles and the microtubules. But you know, centrioles are these mega barrels of microtubules that are much larger. They actually detect photons. I remember I wrote a book in the 80s about microtubules, and there was a chapter about centrioles detecting gravity. And I found two theories that were interesting. One was that it was the centriole was a gyroscope. It actually maybe not rotated around and around, but maybe back and forth, and could act as a spinning gyroscope or something like that. But I don't know. I do know that the stuff about bird navigation and radical ion pairs could be explained by the cilia in the eyes too. I'm not, a different type of quantum effect. I don't know. So in other words, how birds detect the Earth's magnetic field rather than this radical ion pair. It could be microtubule, or it could be radical ion pairs in a microtubule and something else. I think actually that the cilia, which are in the front of the eye, and all the photons have to go through the cilia, which does detect photons in other systems, to get to the rhodopsin in the back. And because of the helical geometry, the cilia might be extracting quantum properties, angular momentum, or orbital momentum, and getting it into the system other than through the optic nerves. There are these Mouthner cells and other glial cells that could. So we may be detecting information from the environment that way, more than meets the eye, more than actually gets to the rhodopsin. The cilia, every primitive ectoderm, or every primitive visual system. I wrote a paper about this a long time ago. Every primitive visual system starts with cilia, a ciliated ectoderm, and then the eye cups are all cilia. And Albrecht Buehler showed that centrioles, which are essentially the same structure, can capture and detect photons. And every rod and cone cell in all of our eyes have these cilia, and every photon that reaches the rhodopsin to cause our vision goes through the cilia, and it could be that they're detecting more quantum information. So maybe we can detect magnetic fields without realizing it. I'm not sure how we'd know that.
Okay, thank you.
Okay.
Okay, Sophia?
Hi.
Hi.
I emailed you I think a couple of weeks ago. I am an undergraduate at UT Austin, and
Oh, I saw that. I probably didn't respond yet.
you responded to say that I should go to this conference, and that we'll stay in touch and everything. And it's an honor to meet you. It's an honor to meet you.
Thank you.
And I am really interested in this subject, and I am wondering what I can do for a PhD. How can I get started to research really about quantum consciousness and your Orch OR theory?
Well, thank you. I don't know, it touches on so many different fields. You could be, chemistry, physics, medicine, biology, quantum optics. So I told you to come to this talk. Is that what I did? And here
Yes.
you are. we're going to have a big conference, the Science of Consciousness conference. We do it every 2 years, and it's coming up in April. And Anirban will be there with a whole lot of stuff about his stuff. And so if you can come to that, either live or online, and just keep studying and follow what interests you. That's what I did. What interests you, and if this is what interests you. So, but your question is which field, and that's a tough one. Paige could probably tell you what fields are getting into quantum biology more than others at this point.
Well, I can tell you that, I took a roundabout path, and you end up where you're headed, whichever path you took. I started in civil engineering, so I'm nowhere near that now. My
Yes.
bachelor's, yeah.
I'm what you were talking about, like with the graph of like that human beings are a lot more higher up in the tubular theory. I found that really interesting. And do you think that can explain why we are so complex and sophisticated as a species, because we perform all of these at a much higher scale than other organisms?
I think we're more conscious, if depending, at least in our theory, we can quantify consciousness by the frequency of conscious events. So, and that's based on the number of tubulins. So we have a lot more, maybe aliens have way more than we do, who knows? But that's one way to quantify it. So yeah, we're more conscious, we're probably smarter in some ways at least. And whether we feel more, I don't know. Feelings are part of consciousness, but maybe different.
And I have another question. Do you take undergraduate researchers?
Not at this time. I have an undergraduate student who's helping me with this megahertz EEG, but he's a U of A student. Check back with me. I was going to be teaching a class next semester, but turns out I'm not. I'll probably do it the following year. But we have a lot of activities at our university, and we're starting a program, educational program in consciousness studies. We have an undergraduate minor in consciousness studies. And as I said, I was supposed to teach a class, but they gave me another year. So because I got to work a little bit more in the hospital because, well, that's another story. But keep an eye on the University of Arizona's consciousness studies educational program, because that could be useful regardless of which area of consciousness studies you go into.
And is there any universities in the world or anything where I could do research as an undergrad in kind of the quantum consciousness model?
I don't know. Maybe at ours in another year or two. I mean, if we get some of these projects going. So keep an eye on University of Arizona. But you're UT Austin now. That's a good school.
Mm-hmm.
And you're an undergraduate?
Yes, I'm a third-year undergraduate.
And what are you majoring in?
Anthropology, but I'm switching to neuroscience.
Pardon me?
Anthropology, but I'm switching to neuroscience.
Anthropology switching to neuroscience. Okay, we have a talk by an anthropologist at the conference, actually, from Stanford, Tanya Lerman. She's going to talk about historical things like history of psychedelics, history of hearing voices, Julian James's ideas, and stuff like that. So, I mean, even anthropology is related to this. But the neuroscience is good, but they tend to think of neurons as simple on-off switches.
Yes.
And you go to the Society for Neuroscience conference. There's 30,000 people there, and nobody's talking about consciousness. They're so deep into their cubby holes and they're down the rabbit holes that they don't see the forest for the trees. So, I used to go, and it got depressing actually, because all this stuff that's so specialized, and nobody's talking about the big picture.
So would physics or biophysics be something that is more akin to this?
Maybe. Yeah, probably more than I mean, we need neuroscience. We need people in neuroscience that with this, inclination to look at quantum effects and microtubules. But what I'm telling you is that right now, neuroscience is pretty negative about this. As Paige or somebody said, that, they criticize us, but it's the emperor's new clothes. They don't have anything better. In fact, like I said before, we have way more evidence for our theory, but it hasn't gotten through. We're way outnumbered. Let's put it that way. So it's encouraging to hear young, bright people like you getting interested.
Yes, I'm very interested, and it's an honor to meet you. And I just want to do this because I found a passion, in this theory, and it was such an honor to see you present the theory, because I'm a firm believer, in this theory, and I really appreciate you looking into this.
Thank you.
Thanks
I'll
so
answer.
much, Sophia.
Yeah, I'll answer your emails.
Yeah, so I know we've already kept you an hour past the time. Just thank you so much, Stuart. This is really a fascinating presentation. We had such good attendance, such good feedback. There is a hand up if we want to take one more.
One more.
So
Ricardo?
I'm sorry. Yeah, do you hear me fine?
Yes.
Yes.
Okay, sorry. Right now I'm in a train station. I had to take a train. So I just wanted to ask if you think that this theory could be in some way correlated to the neuronal transmission in the brain, like an electric. There could be a link between this microtubule theory and neuronal transmission, electrical transmission in the brain. Like there could be a point in which the information that's present in the microtubule can affect and influence the neurophysiological transmission of input in the neurons.
Yeah, well, somebody asked about synapses, but there's also this ephaptic transmission, which is electromagnetic interaction that doesn't need to be quantum entanglement, but just electromagnetic. And Anirban has a paper 2015 in the Journal of Physiology, which is a mainstream physiology journal. And he made these probes. So he had a neural network of a group of neurons and, with integrate and fire, all that. And he made this array of these 8x8 64 nanoprobes, dielectric resonance probes that can measure megahertz and gigahertz. And he put the neural net in the middle, made a sandwich out of it, and could see where you got megahertz and gigahertz, exactly where the neurons were. And what he found was that the megahertz and the gigahertz in the dendrites and soma of a neuron correlated with firing in that neuron and even other neurons better than did membrane integrate and fire potential. So, if neurons are purely algorithmic, we're not going to have consciousness, we're not going to have free will, we're not going to have creativity. It'd just be like a computer. So you need a mechanism to override algorithmic behavior. And they did a study years ago in cats and showed that in a pyramidal cell of an awake cat, that there was a deviation from Hodgkin-Huxley behavior. And that's exactly where you want consciousness to come in. So in other words, you're doing something automatically by practice, walking and thinking about something else. Then all of a sudden something happens, your consciousness takes over. Well, they showed that the membrane threshold changes from spike to spike. So it can't be coming from outside, it has to be coming from inside. Otherwise you'd see it in the membrane. And the functional membrane potential. So it has to be coming from inside. And that's, I think, what the microtubules do. And they could be directly communicating with microtubules in other cells by ephaptic transmission or possibly by quantum entanglement. So yes, they can influence other activity, synaptic and other and non-synaptic activity in nearby and also non-locally by entanglement. throughout the brain. I think we kind of need quantum coherence in the brain, even for a zero phase lag gamma synchrony, brain-wide gamma synchrony. I think you need that. So I think all this megahertz and gigahertz is going to be very, very important.
All right.
Okay, thank you
fantastic.
very much.
Thank you so much, Stuart, and thank you for spending so much time with us today. This is really a fantastic presentation.
Thanks, Paige. Thanks for inviting me. Thank you all for being here. Wherever you are, have a great day.
It was great to meet you.
You
Bye.
too. Bye-bye. Hi, bye-bye.
Bye.